Hello everyone! In this presentation, I would like to talk to you about a very recent and interesting research in the field of biophotonics. But first, let me talk about biophotonics. It is a mix of biology and photonics where photons are used to detect biological materials. Fiber optics are now the rave in biophotonics. This is used in a lot of medical areas such as imaging, diagnostics, and monitoring, and even phototherapy, where an example uses photons to zap the tumors inside the body without surgery. However, there is a limited light penetration inside a biological body, so the usual fiber optics cannot be fully optimized. In this research, a group of medical doctors in the Harvard Medical School have invented the so-called light-guiding hydrogel optic fiber, where instead of the usual glass, a polymer is used. So how do these fibers work? A certain signal from the light source will deliver the excited photons into the fiber. There are various light-sensitive molecules inside our body that can easily communicate with light. In this invention, a polymer called PEG or polyethylene glycol was used instead of glass. In an optical fiber, there is a core-clad structure. The core was fabricated using platinum-cured silicone as the mold. The PEG is then injected into the tube and photocross-linked by exposure to UV. The clad was formed by dipping the core to an alginate solution until multilayers form. The fiber's properties were then characterized using light coupling and/or embedded between the porcine tissues ex vivo. Since the applications of this optical fiber is on the cellular level, this hydrogel fiber was also tested on a mouse. 2 hydrogels were implanted into the mouse where the fiber delivered excitation light at 560 and 640 nanometers, while the other fiber collected diffused light through the tissue. Why is this then a better alternative than the usual optic fiber? Obviously, a hydrogel has greater flexibility than glass. Therefore, it will sustain better in the pressure inside our biological bodies. Also, it has a transparent property which is also important for better light efficiency. Another advantage of this hydrogel fiber is the minimal light scattering loss experienced. As seen in the image, the 2 different fibers were implanted into the rats, and results showed a 40-fold increase in the illumination area with the light guiding scaffold. Since it's a polymer and it has a great diffusion property, it can be injected with functional molecules such as drugs that can be released for therapy in the cells, which will greatly help. It sustains and delivers the molecules properly as seen in the figures. One of the most highlighted advantages of this invention was of its biocompatibility. Because of that, it retains longer viability inside the cell compared to just a simple optic fiber. Results also showed that there were no major cell infiltrations that happened, which proves it is better. Another advantage of this is the result of a higher collection efficiency of the hydrogel fiber than the original fiber. By injecting dye to both fibers, the amount of fluorescence light collected was measured over varying length. In the 4 cm long hydrogel, there was almost an 80-fold increase compared to that of the simple fiber. The patent for this invention was filed by 2 of the inventors who were part of the team that published the paper. The paper was published during 2015, but the patent was already filed during 2014. However, the patent was only approved in 2017. This shows that there is a very high competition in terms of the optic fiber area. The patent includes 21 claims and 13 drawing sheets. After reading and researching about the hydrogel fiber optics, I got so excited about further applications that could be done using this invention. I thought of drug delivery as one of its applications. In the long shot, maybe we could isolate zapping the cancer cells using this drug delivery system. However, in the more realistic sense, it could easily be used in wound healing or scaffold building inside our bodies, where the molecules needed for these mechanisms to finish faster are delivered with optic fiber. In the area of diabetes, insulin could be delivered through this optic fiber rather than the usual skin injection method, which is more hassle and takes a longer time to be processed compared to the direct delivery of insulin to the organs. This ends my innovation presentation. I am Stephany Esguerra. Thank you for listening.